recording, screen sharing. And let me close this. I'm really grinding right now and I can't have any sound for this video.
So I have to slow down.
I don't think you need to slow down.
I'll try to not fumble as much. I'll be doing.
Okay, ready, set, go. Okay, Isabel. I don't see you. No, perfect. Hello, our names are Nicole, Kaylin, Leah, and Isabel, and today we're going to talk about a MATLAB program that we have created for segmentation of brain tumors in MRI. So brain tumors result from abnormal growth of cells in the brain, and they can appear in any part of the brain and skull. They're extremely dangerous, so it is important to identify them quickly. So radiologists use MRI to determine whether there is a tumor present or not. So, but this job can be very hard and overwhelming. So our team has decided to create a program in order to solve this problem. So the goal of our study is to use a box which steps through the image combined with color detection on MATLAB using the MRI modality to detect the presence of glioma tumors, meningioma tumors, or no tumors at all.
So the approach is to identify tumors by segmenting high-density areas of white pixels and highlighting them by changing them to the color red. We chose to do this because based on MRI images, the abnormal tissue seems to be brighter than the surrounding brain tissue. To test this, we will assume two main things. The tumor tissue will be uniform in color, and we'll be forcing each image to be 300 by 300 pixels. With these assumptions in mind, we hypothesize that if the identified pixel is brighter than the healthy normal tissue, we will be able to identify as tumor tissue.
Now let's step through the code. First, read the image of interest, then resize it to be a uniform 300 by 300 pixels, and find N. N is the number of boxes that we want to examine. A box is a 50 by 50 pixel area of the image that will be examined, and the boxes are allowed to overlap. Next, we created an empty vector whose length is the number of boxes. Each index in this vector represents an individual box, and the values within this vector represents the number of identified white pixels within that particular box. So for instance, if num pixels of index 1 is 45, that means that there are 45 identified white pixels within box 1. How did we do this? First, we iterated through a for loop at an interval of 30 pixels for columns and rows. And then we went through another for loop that iterated through the 50 by 50 pixel box we're examining. And if a pixel within this box is white, then we count and save that pixel. Then we create a new vector that identifies boxes with the most density of white pixels and step through that box of white pixels, coloring them red, as shown in this demo. Now let's take a look at some of our other results.
So here are some results for the meningioma tumors that we examined. In the top left, you can see that the full tumor was identified. In the top right, you can see that the full tumor was identified with some of the brain border segmented as well. And that's probably because the border is very thick in that MRI. In the bottom left, you can see that the tumor was partially identified. That's either because the box we used in the code didn't capture it all, or the tumor pixels were not white enough. Here are 3 glioma results from our code. Each one of these examples show partial tumor segmentation, but normal tissue was segmented as well, which was one of the challenges with our code. For our results, it is evident that our code led to different outputs for glioma and meningioma tumors. For the glioma tumors, no image was segmented in an isolated fashion. Also, in around 56% of the glioma MRIs, a tumor was partially detected. On the other hand, with the meningioma dataset, our code worked better. Our code highlighted the correct tumor region in all of the images. The only difficulty was that in about 50% of the images, there was significant false identification of tumor.
In conclusion, our hypothesis was incorrect. The code was not able to accurately segment both glioma and meningioma tumors. Our initial assumptions were also invalid. Tumor tissue is not always a brighter white, and by altering the dimensions of the image, we affected the original aspect ratio, which could have further led to misidentification. To improve the functionality of this project, future iterations could include edge detection in tandem with color detection, changing the parameters chosen, or 3, utilizing machine learning. In the end, a tumor segmentation tool would be very beneficial. Thank you for listening. This was the MatLab Girlies.
You guys rock. 4 minutes and 36 seconds.
Yay.
Finally. Okay,
Yeah, great.
let's
I think
download
that
these
it was
recordings.
great.
Okay,
I
so all you have to do-
I was